I'm going to start off by asking you guys a question. When you guys think of nicotine, do you associate a positive disposition towards it or a negative? Before you answer that, I'm going to re-ask you the question, this time in a more scientific manner. When you guys think of nicotine, do you associate it with stimulating cell growth or inhibiting cell growth? Well, the basis of my experiment is focused on proving if nicotine acts as a stimulant or if it acts as an inhibitor. Initially, when I came into this experiment, I kind of had a post-hypothesis almost, or a pre-hypothesis I should say, where I thought nicotine is bad for the body, so it acts like a toxin and it's going to kill the cells that it's with. So initially I came into this believing already that my pre-hypothesis would be nicotine acts as a suppressant towards cell growth. So let's get into what I'm going to be talking about today. First, I'm going to talk about the practice of sterility. So most of this is going to focus around the methods and then a little bit about the results. I can't really give you a preview without giving you actually what happened, and I'll talk about that later. So start off with the practice of sterility is key in this experiment. You have to be able to grow and maintain your Jurkat cell line, and then I'll talk about what Jurkats are, because, I mean, not everybody knows. And then, the dilution, during this experiment, not just one concentration of nicotine was used. There was multiple dilutions made to test the most efficient concentration to be used. To see if inhibition or if in fact it was the cells were being stimulated in growth. So multiple dilutions were used, and during this the test was conducted 3 times just to make sure maybe that we didn't get an outlier or that something may have went wrong with one experiment, but it was just to kind of get a more accurate reading. And then interpreting the results, I'll cover that later too as well. So now I'll get into why I have the thesis that I have, and that is initially I had the thought that, like I said earlier, my pre-hypothesis that Jurkat cells were going to be inhibited due to the toxicity of nicotine. But after reading some articles, I found one article that said nicotine stimulates lung cancer cell growth. And so Jurkat cells are cancer cells. They're just cancer cells from a boy who had lymphoma. So I kind of associated these cells they have some commonality. They're both cells. So, scratch that last part. So, I looked at the research done by Ying Zhang, Jeffrey D. Ritzenthaler, Jesse Roman, and Shuwei Han, and used that kind of to base my thesis that Jurkat cells, I couldn't come in with a thesis now that Jurkat cells inhibit only. I had to come in with a thesis that maybe Jurkat cells either inhibit or they may potentially stimulate cell growth. So now I have my new thesis. The methods, like I said earlier, sterility. Sterility was key in this entire experiment. Everything you did had to be sterile. Everything. Otherwise, you were introducing outside objects into your Jurkat flask, and thus that could have skewed your entire data. So throughout this whole thing, it was all done under a fume hood with sterile objects. When growing Jurkat cells, you have to reflask them weekly, and what I mean by this is you would take a cell count using a hemocytometer initially every week of your flask of Jurkat cells to get a count as to how many cells you have in your Jurkat media. And what you do then is introduce the same Jurkat cells. You pick an amount from that and put them back into a new flask, and so you reseed the flask essentially, and you keep the cell line growing and growing, so therefore they don't reach their maximum populace and just plateau and you have a bunch of dead cells at the end of it. You're initially keeping the cells alive, keeping fresh cells flowing. So that's the first part of this method. So on top of growing Jurkat cells, I had to also make dilutions for the nicotine. And so what I did was I made 1/10 dilution, a 1/100th dilution, each dilution building off of the other one, all the way up to a 1/1 millionth dilution. And so all these were put into a well, like individual wells, each dilution, and the dilutions also consisted of the cells, but the amount of cells used in each was constant. And there was also a control in the group that only consisted of cells. So we had something to compare to see if cells were being inhibited or if they were being stimulated. So the next part was after putting these dilutions through into a well, an absorbance test was done on these. So what I did was I put them into an absorbance reading machine where it tested to see how many cells were alive at the end after being introduced to nicotine. But I can't tell you the findings yet, so that'll be later. Now, the findings, later, surprise. So the findings, I found that that this is where kind of my thesis was either one way or the other, or it disproved or proved my pre-thesis or hypothesis. So if we look at the data that I have here from the 3 tests that I did, you can kind of see a general flow and general trend that's increasing and then plateauing. So we have the control here, and it's a little bit lower, and then we have the 1/10 dilution, which increases in absorbance reading, and then the 1/100th, which increases just a little bit more, but there's a more significant increase between. This is the most significant increase that we see here, and then the rest kind of just slowly increase a little bit more, plateauing almost. And this happens across all 3 tests. So, what can we formulate from this? Well, I can't say right now, but just think about what you've seen here and just kind of think about it for now. So now we'll jump into some analysis and conclusion. So now, based on what I've shown you, we've seen that the absorbance readings have all increased after the control, and so we can deduce that nicotine does act as a stimulant and not as an inhibitor, and that the most efficient concentration of nicotine is actually between the ranges of the 1/10 dilution and the 1/100th dilution, based on the fact that the amount of increase from the 1/10 to the 1/100th was greatly significant. compared to the later dilutions where it mostly plateaus and nicotine isn't making as much of a difference. So some discrepancies in this experiment are that most of the measurements done were used by doing a pipette, which can be accurate, cannot be accurate. I mean, they're not always 100%, and it's just based on how proper you do your technique too. And I'll confess, I may have messed up a little bit, but I think it should be also stated that based on my results, we kind of see the same thing happening every time, and it looks similar in all three tests, that this probably wasn't actually that big of a deal, and that for the most part my method was strong and little to no weaknesses were found, unless I can't think of any actually. So, not to sound cocky, sorry. So some future research that might be done on this is, now that I know that nicotine stimulates cell growth, I kind of want to know how. What are the mechanisms behind nicotine being able to stimulate cell growth? Here I'd been thinking it's a toxin all this time, but it also makes sense. I guess I never thought of Jurkat cells being cancer cells, so therefore nicotine is inducing cell growth of something bad. So therefore this kind of makes sense, and now I have to look at the mechanism a little bit more. So now we're just going to review kind of what I've found and what I've been talking about today. Nicotine acts as a stimulant towards cell growth, and its most effective concentration ranges from 1/10 dilution to 1/100th dilution. Throughout this whole thing, all three tests showed the same significant data, and that's all I can say about my review. In conclusion, I'm now able to say that nicotine acts as a stimulant confidently, and now the next question I have for myself is looking at how nicotine works in our body and what the mechanism behind it will be. So, thank you.